So the biosynthesis process of penicillin. So how the penicillin it is going to produced. So how the penicillin it is going to produced inside the microorganism. See here. So see exactly the biosynthesis of penicillin. How the penicillin biosynthesis is going to occur. So the first is the L-alpha amino adipic acid. So exactly the L-alpha amino adipic acids. So where the L-cysteine is going to added. So if the L-cysteine is going to added, so there is formation of L-alpha amino adipic cysteine. Then after the L-valine is going to get added, and then the structure that is going to called as L-alpha amino adipic cysteine valine, that is going to called as the tripeptide. And exactly if the cyclase is going to present here, so that is going to form isopenicillin N, isopenicillin N exactly, and the phenyl, then finally that is the phenyl acetyl coenzyme A, and there is releasing of alpha amino adipic acid, alpha amino adipic acid plus coenzyme A, that leads in the presence of enzyme acyl transferase. So that leads with the formation of penicillin G. So as a result, the penicillin G it is going to form here. So the first is the biosynthesis of the penicillin. So exactly, see exactly the alpha amino adipic acid when it is going to combines with the cysteine here. So if they are going to combines with the cysteine, exactly there is formation of amino adipic cysteine, and if the amino adipic cysteine is going to form here, so they are going to combines with the another amino acids, that is the valine here, that leads to the formation of alpha amino adipic cysteine valine, that is the tripeptides. So the cyclic enzyme is going to responsible for the cycling structure, and that is going to called as the isopenicillin N. And if the alpha amino adipic acid and coenzyme is going to remove here from the phenyl acetyl coenzyme A in the presence of acyl transferase enzyme, that leads to the formation of penicillin G exactly. So this is the mechanism how the biosynthesis of the penicillin it is going to occur. Then after I am going to explain about the production process of penicillin. So what are the ingredients that we are going to add for the production of penicillin? See here. Then the next is the Then the next is the production process of penicillin. So the production process of the penicillin is that, see here, what is the production process? So the production process is that it is mainly a aerobic process. It is a aerobic process, exactly, and therefore the oxygen supply is very much essential. See here, so the oxygen supply is very much essential for the penicillin production, and exactly here the aeration rate, that is the aeration rate, is equal to 0.5, and see here the pH that is going to maintained, the pH that is going to maintained is 6.5, and the optimum temperature for the penicillin production, and the optimum temperature which is going to used for the penicillin production, that is a 25 to 27 degrees centigrade, and exactly the penicillin production is carried out by submerged process. Penicillin production carried out by submerged process. So exactly, so this is the most important. And what are the composition of the media? So if you can see here the composition of the media, so the composition of the media, that is going to consist of carbon source lactose, that is the carbon source, it is going to consist of lactose, then the nitrogen source, that is the yeast extract, the nitrogen source, that is the yeast extract, and phenyl acetic acid as a precursor for penicillin synthesis. The phenyl acetic acid that is going to the precursor for penicillin synthesis. Then exactly, so this is the most important point that is going to be, and the overproduction phase, the penicillin production phase is going to require, see here, the penicillin production phase is going to consist of 150 to 180 hours. So it is going to consist of 150 to 180 hours. So see here, the first I am going to explain, so penicillin production process. So the first point is that it is completely a aerobic process, है ना? So it is completely a aerobic process here, and oxygen supply is very much essential for the penicillin production, and the aeration rate means the oxygen supply is very much important. pH is going to maintain 6.5 2.5, the temperature is going to require here and the 25 to 27 degree centigrade, and the production process through which the penicillins are going to produced here, so that is going to called as the submerged process. And here what is the composition of the media? So if you can see the composition of the media, the first is the carbon source, so that is the lactose. So the carbon source that is going to utilize here is the lactose. Then the next is the nitrogen source, which is going to consist of an yeast extract. So the nitrogen source is going to consist of an yeast extract, and the phenyl acetic acids, that is going to the precursor of penicillin synthesis, and the penicillin production phase is mainly going to consist of 150 to 180 hours exactly. So this is the production process. So what are the conditions that is going to required for the production of penicillin, and what is the composition of the media through which the penicillins are going to produce inside the bacterial cell, inside the fungal cells. So what are the carbon source, nitrogen source, and what are the supplementary, that is a phenyl acetic acids, which is going to added for the production of penicillin. Then after I am going to explain the graph. See here, then the next is the graph. So the two graph is going to important that I am going to explain here. The first is the, see here, this is the penicillin production in relation to substrate utilization and biomass. So this is the concentration, and here this is the time in hours exactly. Then here is the lactose, then this is the penicillin, this is the biomass, this is the ammonia. So see here exactly, so this is going to called as the lactose here. This is the lactose, this is the penicillin here, this is the biomass, this is the ammonia. So this is the graph that is going to the penicillin production. See here, that is the figure, that is the penicillin production relation to substrate utilization, relation to the substrate utilization and biomass. So exactly see here, so the first point is that this is the lactose, it's the carbon source is going to down, this is the penicillin, this is the biomass, and the nitrogen concentration is also going to get decreases. So that this is the carbon and the nitrogen source is going to get decreases, and this is the biomass, that is a lag log stationary, and this is the production of penicillin here. So here, so this is the graph which are going to explain the penicillin production in relation to the substrate utilization and how the number of microorganisms is going to increase, that is the biomass exactly. Then after I am going to explain other graph. Then see here, exactly. Then the next part, then the penicillin production in relation to the feeding of sugar. See here, this is the concentration here, that is going to called as the concentration here, and this is the time which is going to associated in the hours. So the first here, see, this is the penicillin, this is the dissolved oxygen, this is the sugar feeding, and this is the CO2. So exactly, see here, this is the penicillin production, this is the penicillin production here, this is the dissolved oxygen, this is the carbon dioxide, and that is going to called as the sugar feeding. So exactly, this is the graph which is going to explain about the penicillin production, that is the penicillin production in relation to continuous feeding of sugar O2 utilization and CO2 formation. So exactly, this is the most important step. So how the penicillin production is going to related to the continuous feeding of the sugar or the oxygen utilization or the CO2 formation. So exactly, this is the lab here, है ना? So this is the graph here. So where it is going to indicates how the dissolved oxygen and the sugar feeding and the carbon dioxide is going to associated with the production of penicillin. Exactly. Then after I am going to explain about the recovery of the penicillin. So how we can going to recover the penicillin here. So the next step, that is going to called as the recovery of penicillin. Then the recovery of penicillin. See here, how we can going to recover the penicillin. The recovery of penicillin. So as the fermentation is going to completed here, see exactly the recovery of penicillin. So penicillin is recovered by the solvent penicillin is going to recovered by the solvent, that is the N-butyl acetate and methyl ketone extraction, that is the N-butyl acetate and methyl ketone extraction. So that is the two most important solvents which is going to used here. And finally the penicillin can be removed. So the next is the activated carbon is going to remove impurities. Activated carbon is going to used to remove impurities, and finally the penicillin can be recovered. Penicillin can be recovered by adding potassium or sodium acetate. By adding potassium or sodium acetate. है ना? Exactly, see here. Then finally the yield of penicillin is 90%. Then the penicillins are going to crystallize here. And used in dosage form. And used in dosage form. So just write down exactly how the penicillin it is going to recover here. Then the next point, see here. Then the next antibiotic. So first, if you can see, that is the penicillin that is going to recovered by the solvent, that is the butyl acetate and the methyl ketone, and the activated carbon that is going to used to remove the impurities. Then the penicillins are going to recovered by the potassium and sodium acetates, and they are going to crystallize, that is going to used in the dosage form. Then exactly the next antibiotic production. First write down the recovery.